Thank you very much to both of you for a very kind introduction. It's an honor for me to be here today. It's a really, I actually feel a little out of my depth because we haven't done a great deal of valve work in the last 4 or 5 years. But I know that you want to know the latest about endothelial phenotype as it may apply to the valve. And so what I'm going to do today is try and look forward and to the present in terms of endothelium, particularly in the arterial circulation, as it relates to hemodynamic environment, and then move that across to the valve and tell you a few interesting things we've done in the valve, and what I think will be a way to move forward in a lot of mechanistic studies concerning the endothelium and its hemodynamic environment, which I hope will lead to similar large amounts of papers that are coming out similar to the arterial circulation. So with that, bear with me, and I hope it will make relevance sense to you. Now, for many years, we've been very heavily involved in the focal and regional origin of atherosclerosis. The equivalent, of course, in the valve is the aortic valve. In the aortic valve is the sclerosis, inflammation, as well as calcification that accompanies aortic valve disease. And we can add to these locations which are very prone to atherogenesis. All of them are related to very complex hemodynamic blood flow. And that's true also of the valve. And it's also true of the coronary arteries, which of course are a major clinical problem. So just a few examples of the kind of flow disturbance that one picks up in the large arterial circulation, shown here. In the aortic arch is a very strong predictor of atherogenesis, and you see the separation of flow. Obviously, you have to look at certain regions of the cell cycle to see this. It's principally post-systole in the diastolic portion of the cardiac cycle. And the endothelial cells are recipients of two major mechanisms which influences their biology where the flow is concerned. One of them is the biomechanics of the forces in the blood, largely modeled on Newtonian characteristics, even though the blood is non-Newtonian, but it actually is a reasonable approximation for a lot of the studies. And secondly, transport of chemicals, both produced in the blood, and also coming out of the vessel wall, which everybody forgets about, which are actually in some kind of steady state or alternating pattern of concentration related to the hemodynamic flow. And this is a very difficult area actually to tear apart. There is strong evidence that the hemodynamic biomechanics is definitely playing a very dominant role, but in some circumstances I think that the particularly reactive oxygen species, as we've heard about this morning, the transport issues I think loom large and will loom larger as more research goes on. So the carotid bifurcation is another classic example of atherogenesis, focality, and again you see from beautiful work from David Steinman the flow separation that occurs in the carotid sinus specifically as a function of the just after peak systole, and this collapses with a lot of reversal, multi-directionality of flow, and again a predisposition to atherogenesis. And this atherosusceptibility occurs in the normal vessel. You can take a young animal and find it. And the question is, do the endothelial cells lining these regions, which in most places throughout the circulation, arterial circulation, align in the direction of the flow, are they in fact a different phenotype? And in fact, if you look at these regions, arch, carotid bifurcation, you will find regions of what could be called stagnation points. They're not really stagnant because they move back and forth, but the endothelial morphology actually dictates, this polygonal morphology dictates the regions where the flow and the biomechanics are radically different from nearby flow directionality, undisturbed flow. So we call this undisturbed flow up here, and for want of a better word, we call this disturbed flow because we don't know what else to call it. So this has led to the idea some years ago that there is an atherosusceptible endothelium, which is, or if you like, inflammation susceptible, if you wish to view it that way, associated with even normal tissue, where the phenotype subpopulation of endothelial cells is different than in other locations. Now, the approach that we've taken is a little bit different, and of course, we very foolishly found it very challenging as well. We use a large pig as our model. And in actual fact, we start with the in vivo because I spent 25 years doing reductionist studies and then became a little perturbed at the disjointedness of a lot of high-throughput genomics I was doing on the cells in culture, that they in fact reflected what was going on in vivo. So I turned it on its head and said, okay, let's see what nature does. And this is a principle which I think is yielding rather interesting and sometimes unpalatable truths in terms of the phenotype mechanisms that are going on. But it has certainly yielded a bonanza of interesting data for us. We start by isolating the endothelial cells from these specific locations. There are various techniques, it's all published. And we can then isolate whatever we want, nucleic acid, cells themselves for protein, RNA, DNA, and so on. And then we can do next generation sequencing or microarrays, whichever you can afford to do. And from that, you move on to the really important bioinformatic analyses and analyze the systems biology of the steady state condition of those cells in that animal at that site when you took them. And so it becomes a site-specific analysis. And the site-specific differential expression, a whole slew of RNA transcript levels, messenger RNA, so gene expression, is an enormously powerful multiple of independent single molecule approaches. We can do pathway analyses, which are with companies like Ingenuity, are very sophisticated and of use as long as you keep your head switched on as to how well they reflect the literature and how some of the literature is flawed. But then again, you can go on to validate with QRT-PCR and Western blot and immunohistochemistry, as we've seen this morning in various cases. From this, we get really a survey of what's going on in vivo. And then the interesting things happen because from the systems biology, we do the reductionist experiments. And there we are setting up flow conditions which try to simulate what is going on in vivo for the particular problem that we want to address. And if we find from that, because we have control, we can dissect it very, very extensively and discover and confirm new mechanisms. And then we look for evidence for these mechanisms in vivo. Now, the reductionist experiments often throw up information which you didn't realize in a pathway existed, but you only were led there because of the in vivo. And it's important, therefore, to go back in vivo and check indeed whether the new discoveries are actually occurring back in the animal. Now, most studies start at this point, and in fact, I did for many years, and it's perfectly fine to set up a hypothesis, do the reductionist experiments, get a mechanism, and then look for some evidence supporting that mechanism in vivo. We've tried to, in many cases, to complete the cycle, starting with in vivo and ending with in vivo, and have been fairly successful in finding some new things. Now, a couple of examples of issues which come up, and I mean, they're very extensive. There's a very large literature on this, including from our own lab. Really began back in 2004 when Tony Passerini was in the lab. And basically, an example is that one finds that the genes are up and down regulated in the disturbed flow region of the aortic arch compared with adjacent regions where the flow is not disturbed, in patterns such as clusters of genes. And this example shows what would be a pro-inflammatory environment, both leading into an NF-kappa B pathway, which you've heard about today. The up and down regulation points to pro-inflammatory environment. Yet, when you look, we don't see actual inflammatory activation in the normal endothelial cells under these conditions. And if you look at another segment of the systems biology, you find a very strong antioxidative profile in the normal endothelial cell, even in disturbed flow, which possibly attenuates the inflammatory response. And so the consequences of activation of NF-kappa B from a cytosolic to a nuclear location doesn't occur. And indeed, you don't find it in these locations. And the adhesion molecule expression, which often results from this kind of activation, you heard a little bit from Dr. Farrar this morning from the Butcher group, when you hit with TNF-alpha or something and drive this, you really get very strong adhesion molecule expression and you get inflammation. But this kind of balance exists in the same cells only by going at it with a high throughput approach. And what the net effect is a very low level of inflammatory phenotype in this cell. And there are many examples in coagulation biology and in lipid metabolism and so on, which we've uncovered in this relationship. A few are shown here. We discovered that ER stress is a consequence of disturbed flow wherever we look. It's a way in which the cell adapts to stresses, including endoplasmic reticulum stress, to calibrate the biosynthetic pathways of protein biosynthesis, the pathways in the endoplasmic reticulum, and adapt the chaperones to be able to handle an overload when the cell is stressed. And this is 7 or 8 papers now we've published in this area, which is holding up very well. So you already have, in a disturbed flow region, an endothelial cell whose phenotype is stressed, and stressed by, but it's adapted to that stress and functions normally. The reactive oxygen species balance is balanced by the antioxidant protection in these studies. The inflammation, anti-inflammatory pathways I just showed you, and lipid metabolism gets balanced. There are responses which allow for less insudation, or less, I should say, not insudation necessarily, but exchange of lipid to the endothelial surface. So the regulation of gene and protein expression can be demonstrated at the genomic through high throughput screening, the epigenomic and epigenetic through high throughput screening, and translational and post-translational levels. And all of these have now been demonstrated. And these areas really, I think, are right to expand in studies of the aortic valve and, in fact, the mitral valve research. In addition, we're able to extend to hypercholesterolemia by taking these animals and giving them a systemic insult and seeing what happens to a local effect. So here's a summary, just to finish this first part, on some of the things that we know are going on from this kind of study. We know that this ROS can itself, because of flow disturbance, as we heard this morning, cause biosynthetic load to increase, to induce ER stress, which, if unchecked, will kill the cell. The cell usually adapts normally, and indeed it can result in production of more ROS and actually cause a loop of reinforcement in a negative sense. However, the unfolded protein response stimulates chaperone synthesis, which attenuates this effect. So you end up with this, as I showed you earlier, low-level chronic inflammation. The regulation of this low-level chronic inflammation and factors which may alter it, we know are influenced by up and down regulation of a number of factors. And I'm showing you here not to get into the detail. This is work principally from our group, Xu Chen's group, Hanjun Zhou's group, the Georgia Tech Emory axis, and a lot of other labs as well. And indeed, it's very clear that small non-coding RNAs, microRNAs, play an important role in regulating key molecules, which are the expression of which are up and down regulated in disturbed flow. And eNOS, KLF2, KLF4 are very dominant examples which protect, and if they're down, you lose that protection. And IL-6 and ICAM, VCAM, MCP1, and FKB pathway, all are pro-regulatory, and they're influenced by some of these regulations. Now, microRNAs and other non-coding RNAs are now pretty much viewed as part of the epigenomic architecture, because they take the RNA and destroy it, but the RNA is still made first. So they're regulation of post-transcriptional regulation of expression of these molecules. But it stops translation, so there's no protein. Now, you add the cytokine mix into this, which is typical of any environment where there's an inflammatory event going on. And you layer on top of this the MAPK pathway, junk P38AP1, which enhances the chronic inflammation, analogous to TNF-alpha adding to cells in culture, for example, that you heard this morning. And in addition, there's crosstalk between NF-kappa B and junk and ATF2. And we've shown that post-translationally that protein kinase C zeta, but only zeta, has a very potent effect in terms of mediating this effect. So this is at the transcriptional, the post-transcriptional, translational and post-translational levels that one is dealing with regulation. All of these elements can be applied to the valve endothelium as well, with the appropriate environment. Now, added very recently, and this is from 3 labs, we, I think, managed to get it out first, there's a flow-related epigenomic. We don't know if it's epigenetic because we don't know if it's inheritable yet, but we're working on that. DNA methylation change, which is influenced by disturbed flow. And we demonstrated, this is work of Yizhu Zhang, who's in my lab, a postdoc, a very good postdoc, came out in, I think, April in Circ Research, for a very deep analysis of the mechanism of couple-like factor 4, KLF4, which is highly protective. And it turns out that it can be suppressed in the endothelial cells by DNA methylation of its promoter. And it's a very nice mechanism that he's uncovered there as an example. And we think this is probably a more general mechanism than is realized. And it's a direct response to exposing endothelial cells to disturbed flow in vitro. And this is, again, there's a paper just, I think it's out now, as an emerging topic that I've written, which summarizes the 3 papers that are involved in this observation. This is a hot area, and I think there's some cases to be made for the valve being a classic location for environment affecting gene expression through epigenetics and epigenomics. Now, much of the DNA methylation that goes on is developmental biology and is fixed, but some of it, I think, is somewhat physiological in actually being able to regulate transcription of very important molecules. Just to add to those who aren't quite aware of it, KLF4 and KLF2 are transcription factors, and that one of their principal targets is NOS3, or endothelial NOS. And so endothelial NOS, although not directly affected by DNA methylation, we think, Phil Marsden may disagree a little bit at Toronto, but it doesn't seem to be. But eNOS itself is a target of these transcription factors. So you affect these transcription factors, you would potentially influence the expression of eNOS. And here's the idea now that the epigenetics or epigenomics really adds all 3 of the principal mechanisms that we know influence the code. In other words, it's superimposed by other influences upon the code without changing the sequence of the DNA. That is DNA methylation, which influences transcription. The histone code, which wraps around as you know, DNA wraps around histones, proteins, and sometimes it loosens them, allowing other factors to get in there and do their work. In other cases, it's direct consequence of DNA methylation, and these work back and forth. And the third one are the short and long non-coding RNAs, which influence the already transcripted, which influence the transcript messenger RNA already and destroy it or allow it to proceed. So this is an example now where we have all 3 components of disturbed flow. There is evidence in the literature from Xu Chen's group and from Han Jones' group and from us that indeed all 3 of these are now operating as a consequence of the disturbed hemodynamics for the endothelial cell. The consequence of this particular exercise of our paper was that methylation silences genes, stops transcription through promoter methylation on cytosines, and demethylation allows genes to express. And what people, I think, have forgotten is that the methylation is a cyclical process whereby DNA methyltransferases, 1, 3A, 3B in particular, allow for methylation, and TET is implicated through a complicated mechanism in actually degrading or demethylating. And of course, whenever you have a cycle, you have dynamics, and you have an input and an output. And so regulation of these mechanisms, in fact, can influence gene expression. And what we found was that DNMT3A, DNA methyltransferase 3A, was actually recruited to the promoter of KLF4, and through a MEF2-dependent pathway and blocked with anti-methylating enzymes, we were able to demonstrate disturbed flow was directly causing a downregulation of KLF4, and as a consequence, NOS3, thrombomodulin, MCP1, all of them associated with inflammation, pro-inflammatory behavior, were influenced by the downregulation of KLF4. So it's one mechanism influencing the outcome of very important downstream inflammatory molecules. Now, let's move to your real love, the aortic valve, but keep in mind some of the principles that I think I'm trying to outline for the arterial work. Obviously, we've heard tons of this. We all know that the calcification is an actively regulated focal disease. But it's also involved with sclerosis. The two go hand in hand, and the question is how much interplay is going on, or are they actually operating a little bit separately? I'm not clear in my mind that they are necessarily totally hand in hand, even though they're co-temporal in many cases. And I'm assuming that, and this is a tip to the organizers, tip of the hat to the organizers, that everybody has read this rather good review, which has come out recently, which really outlines a great deal of the global biology of the aortic valve. The key thing in the title is the living aortic valve, and it's the dynamics and the mechanisms that really, and the equilibrium of those mechanisms, which probably cause shifts which have become pro-calcific, pro-sclerotic, and at other times may not do so. And I think we're missing the dynamics to a large extent. So let's explore that a bit more. As you know from lovely work from both Ajit and Mike Sachs, defined and modeled very nicely that the two sides of the heart valve, the aortic valve, have very different hemodynamics. So here are the parallels. Here is the aortic side of the valve at two phases of closure and opening of the valve. And here is the ventricularis side, endothelium. And the kinds of forces, and which is the color, and the vectors of those flows are shown. And you can see it's a much more complex hemodynamics environment for the aortic valve. So the endothelial cells on the aortic side experience disturbed flow. Apropos arterial cells. Are they a variation of what we're finding in the arterial system? I think Jonathan Butcher's work, again, comparing arterial endothelial cells in culture systems with valve cells, again, was an eye-opener for differences and similarities between these subpopulations. But let's look for similarities. Those on the ventricular side experience high shear, mostly unidirectional and fairly undisturbed flow. So we really began to look at site-specific endothelial phenotypes over various length scales in the valve back in about 2005 when the Canada chair professor who's here today at the University of Toronto, Craig Simmons, was a postdoc in the lab. And what Craig did was the first in vivo study of isolation of endothelial cells from each side of the aortic valve, confirming their purity and so on, confirming that we could get enough RNA of very good purity and good enough amounts to do high throughput analysis. And this is an old scheme. Now we would sequence instead of doing microarray, but essentially this is the idea that you can amplify and then you can look at the genomic profile. And just to give you a couple of examples to make one single point, if you look at two examples of higher and lower expression, they tend to favor a susceptibility of the aortic side of the valve to calcification. Whether these are up or whether they're down tends to fit in with a more susceptibility. And when Craig looked at immunohistochemistry, there's some startling contrasts of a number of them. This is osteoprotegerin, which is fairly protective against calcification, and it's expressed exclusively on the ventricular side. It's totally absent on the aortic side, endothelial cells. This is at higher power. Another example, again, here you see a lot of expression in the interstitial cells as well, but there's a very strong expression of 12-lipoxygenase, again, on the ventricular side and not the aortic side. Both of these molecules would be somewhat protective. So that protection is lost on the aortic side. Again, you could consider that as susceptibility. So the other thing that we followed up with in our small amount of work we've done on the valve was to look at hypercholesterolemia. Initially, the hypothesis was, okay, we now give these animals hypercholesterolemia systemic challenge. We know that it causes certain changes, and I'll show you those in a moment, even after a couple of weeks of hypercholesterolemia. And we should be able to do sidedness because the lesions are not really forming very extensively, and we can still harvest endothelial cells pretty purely from them. And this work was done by Marie Geraghty, who's an MD PhD, who's now Chief of Cardiology at Penn. I'm sorry, Chief of Residence at Penn. She did two analyses in the same study. The effect of hypercholesterolemia on each side, on cells on each side of the valve. So it's, if you like, it's the aortic side only. What's the difference between hypercholesterolemia and normal cholesterolemia in these cohorts? And what's different in the ventricular side between NC and HC? And she did also a comparative study of sidedness within each dietary treatment. And now you may or may not expect these to agree or disagree, but by doing both of them, you actually gain information. It's 1 + 1 = 3 in many respects. And let me show you some of the things which happened. And Magdi, I don't know if the pig is your favorite model, but we were constrained to use the pig for this. What we see at 2 weeks is extensive regional insudation of lipid. At 6 months, we see, as we've seen in earlier pictures, the frank lesions forming on the aortic side. Of course, this is always on the aortic side. The ventricular side is unaffected. And the valve at 6 months has nodules in these animals quite markedly. We also see evidence of calcification. We think it's calcification at 2 weeks. And these are some of the H&E studies, examples, again, always on the aortic side, sometimes associated with some insudation, sometimes some distance from insudation. And bialza and crimson staining, they show up very prominently. Now, again, I don't know whether this is inflammation as well or not, or whether it's a calcific gene repertoire that we should be looking for. But what Marie found in the longitudinal study was that after 2 weeks of hypercholesterolemia, there were a number of genes, a large number of genes, which were changed on the aortic side only. And this is this part of the study. It's the aortic side looking at the effect of hypercholesterolemia. When these were assembled and analyzed by very sophisticated bioinformatics, they showed a net protective effect in terms of gene expression for inflammation and for calcification, as far as we could judge, but particularly concentrating on inflammation. So there's a TGF-beta centric pathway, but the direction of up and down regulation of these genes, which directly influence TGF-beta, were in fact protective and not pro-inflammatory, which is the exact opposite of the hypothesis we went into the experiment with. And so Marie then, well, I'll give you an example. For example, fatty acid binding protein 1 and PPAR gamma are, again, in a complex way, interconnected in a protective way. And indeed, here you see in normal cholesterolemia, you see PPAR gamma, there's very little. FABP1, there's very little, if any. 2 weeks, both of them upregulate on the aortic side and not on the ventricular side. At 6 months, it's still there, which is kind of, we don't really know what to make of it because we kind of expect it to go away and be replaced by frank inflammation. ABCA1 is a global protective effect against hypercholesterolemia, and you see it's even upregulated in the normal state and is enhanced in the hypercholesterolemic state. On the ventricular side of the valve, the same kind of study, as opposed to the aortic side where 1,300 genes were differentially expressed by hypercholesterolemia, only 80 were on the ventricular side. So a vastly different response to hypercholesterolemia. And of the ones that made the most sense in terms of pathway were these G-protein relationships, all of which were upregulated. And again, we haven't taken this very much further, but I think there's an interesting story there. Whether it relates to some kind of protective phenotype on the ventricular side, we just don't know at this point. The other analysis compared sidedness. So under normal cholesterolemic conditions, the sidedness was basically what Craig had done, and we confirmed that. The effect of hypercholesterolemia, however, was again to result in relationships which point to an anti-inflammatory pathway. TNF-alpha is synthesized and secreted by endothelial cells in the valve, and it's a downregulation, blue is downregulation, of TNF, whether we look at it from two separate kinds of analyses. And I won't bore you with all the details, except to say that there is a very clear protective bias to these studies. And so, and again, this is demonstrated, if you ask about the classic molecules, for inflammation. You look at VCAM, ICAM, and other CAMs, and we've been unable to demonstrate a bias to one side of the valve or the other, either in a normal valve, which is where it's very low, or in a hypercholesterolemic valve. And a typical story is this kind of relationship, where you'll find a region which is clearly positive for ICAM, but it's not much else on the aortic side along here. And you find a region which is positive down here, and not much up here. And when you actually do the quantitative analyses, they balance out. They're about a unity. So we can't say the susceptibility or the development of inflammation is because of ICAM, because there's no evidence that it's different on either side at the average measurement of the cells on each side. And the same is true for VCAM. However, what we have found is one CAM which has, in fact, shows a response to hypercholesterolemia, and it's ALCAM, activated leukocyte adhesion molecule, CD166. Very little out there in the literature on it. I'll return to that in a moment. But what you can see is in control normal cholesterolemic, it's really not expressed. But in the valve, it's expressed primarily at the endothelial ends. There's some expression in the interstitium too. And at 6 months of hypercholesterolemia, it still appears to be expressed extensively. And again, these are blow-ups of each of these units. So you can see, even though this looks like there's some expression, when you look at high power, it's not very much at all. Whereas up here, it's quite intense. And you see it very clearly in the endothelium for 2 weeks versus the ventricular side. So now we have at least a molecule that we could work with to see if indeed it bears a relationship to the propensity for inflammatory behavior on the aortic side endothelium. Now, ALCAM CD166, the only papers that are out there, other than some developmental biology papers, are that it clearly is a component of the endothelial cell involved in transendothelial monocyte migration, which you'd expect it to be, given the fact that it's a CAM. That the activated molecule promotes leukocyte trafficking into the central nervous systems across the blood-brain barrier. Again, that's another paper that's out there. So it's getting very much associated with transmigration of monocytes, which is a hallmark, of course, of inflammatory behavior at the functional level. And recently, and this is now why it gets interesting towards epigenetics, is that MIR126-5P is a regulator of ALCAM and another molecule, regulating leukocyte adhesion and transmigration. So now we have a potential mechanism which is epigenetic in nature, which may influence the behavior, the functional behavior of ALCAM. And I think this is an interesting direction which may be applicable to valve biology. So the approach that we've taken is to take, so far, is to take all of the endothelial cells, as much as we can, from either side, to do an analysis which is high throughput, and then to try and figure out what it means for disease-sidedness. But I think we're missing something. And what I think we're missing is the subpopulations of endothelial cells within a given side, because we have so far taken all the cells and averaged out, and we may be missing some of our results may be as an average of potent local effects which get diluted in the approach. And the way to address this and to overcome this limitation is essentially to use laser capture microscopy, which is extremely tedious, but I don't see any other way of doing it, to isolate exactly the cells that you want to analyze and to gather enough from the hot spots, if you like, both in disease and in hypercholesterolemia and other disease modes, as well as the normal state. So cell-to-cell adhesion and inflammation needs to be analyzed at a higher resolution at a different scale. So to summarize, what I've tried to get across to you here, and I apologize for all the arterial work, is that site-specific differential gene expression in susceptible endothelium may represent a chronic adapted state. The cell is always going to try and function. It's got mechanisms to try and protect and to function. And what we're seeing, I think, are the dynamics of equilibration and re-equilibration constantly going on in the cell, which allows it to survive. Reduced to quantitative, it's essentially rate constants for every step of every pathway that the cell uses to function in, which is a daunting task to dissect, but one can be selective. Secondly, whatever the primary causative mechanisms, the endothelium experiences regional, genomic, and epigenomic regulation through a hierarchy, transcriptional, translational, post-translational, of control. And that hierarchy is responsive to hemodynamic change, to disturbed flow of certain characteristics, often with an oscillatory component, with a multidirectional component, and with a magnitude of shear stress. And in fact, some nice work from Penn State indicating it's also frequency responsive as well. So that means that we can analyze and target transcription factors, microRNAs, DNA methylome, and other emerging epigenetic differences. All of this applicable to valve investigation. The third point is that the aortic valve endothelial phenotypes are different on the aortic side from the ventricular side. That is obvious from what I've shown you. And again, we don't fully understand how accurately we're measuring that and exactly what those mechanisms imply in terms of the dynamics of inflammation and calcification. Fourthly, upon systemic challenge, and that's hypercholesterolemia, the local site-specific response of the aortic side is way, way higher than the ventricular side. So the plasticity of phenotype is far greater in response to an insult, a systemic insult, for one side of the valve than the other side of the valve. Terribly important thing to keep in mind. Now, whether that means that this is super protected and that's loss protection, or whether it means that's super susceptible and that's not susceptible, it's a little bit of semantics. The hypercholesteremic challenge results in a paradoxical protective response in the aortic side endothelium. And again, as I said, this could be an averaging effect, and it requires very good focal studies to sort that out. And finally, CAM expression tends to be similar on each side for the usual suspects, but for ALCAM, there may be a story in there because it appears to be inducible on the aortic side only by hypercholesterolemia. So I'll stop there and I'll acknowledge a large number of people, particularly our relationship with the FDA Center for Devices and Radiological Health, just outside of Washington, where we interact. Bill Pritchard did part of his PhD with me when I was at Chicago and runs that lab, and they're fantastic colleagues. This is a team, we go down as a team. The ones with the hooplas are the FDA people, fantastic animal surgeons, radiologists, and we go down and do the harvest with them and gather the tissue as fresh as it possibly can be for our studies with under controlled conditions with the pigs. And a lot of my group, again, gathered together for a gathering at my house fairly recently. Some of them moved on now to faculty jobs or to postdocs, and they're a wonderful group of people. But I'd single out particularly, Yun Fang, who did the microRNA work, and is now at the faculty of the University of Chicago. Elisabetta Manducci, who's a fabulous staff member at the Bioinformatics Center at Penn, who's not just a service person, but a colleague, a scientific colleague. Tony Passerini kicked off a lot of this work. He's now an associate professor at UC Davis. And Mette Chevalik, who uncovered the ER stress story and is finishing a postdoc with Jake Lucis at UCLA and is looking for a job, by the way. And this is all done at the Institute for Medicine and Engineering at Penn. And lest we forget, we stand on the shoulders of giants. Da Vinci in 1490 anticipated the flow disturbance posterior to the aortic valve and drew it in his lab notes. He also wrote his lab notes in Latin, in reverse, mirror image Latin, and he certainly didn't need a protected, encrypted file for his work. Thank you very much.
Thank you very much, Dr. Davies, for such a comprehensive, stimulating talk. This paper is open for discussion. Can I ask a question? Now you've been studying the genomics that at local level, RNA expression and epigenetics. What about inherited factors and regulating genes, either inhibitory or stimulating? And now that we have next generation sequencing, we can look at the whole lot rather than...
Well, again, I'm speaking for the arterial endothelium now. We've done the whole genome methylome, all the DNA methylation sites that are different in disturbed flow region versus an undisturbed flow region. There are 564, which is a small number from such a large, from the whole genome. And they're targeting a number of genes which are almost a wish list for those of us studying atherogenesis. Now, it implies that the epigenetic DNA methylation pathway is active, and it's active in two ways. The way I described, which I think is probably fairly rare, which is that it is pliable or plastic and can respond to an environment and not necessarily inheritable. And the second one is the point you're raising, and that is that the majority of those DMRs, or DNA methylation regions that we find on all the chromosomes, in fact, are inherited during developmental biology and are fixed. And they have prominence amongst the families of genes which are targeted in that analysis are the Hox genes, which of course have a very long history in developmental biology as key superfamily of genes for positional development in the mammal, in all organisms for that matter. So there's an example where I think the epigenetics is not a division, but there is a difference between plastic DNA methylation and fixed DNA methylation, and it'll be very important to make that distinction very carefully in analyzing the importance and the possibility of changing some of these epigenetic control hierarchies, which clearly are influenced by hemodynamics.
Excellent. Can I ask another question while they're coming to the microphone? The endothelial heterogeneity around the body, is that fixed and genetically determined, or is plastic and you can change it at any time?
I think a lot of it has to be genetically determined, or else I think it would be too evolutionarily it would be disadvantaged. Perhaps you could argue it would be an advantage to be plastic, but once the species is established, I really think that a lot of fundamental fixtures remain so from developmental biology, and layered upon those is the epigenomic story, which I think is just opening.
So they remember their origin, the lineage.
Yeah.
Okay. Yes, sir. Can you state your name as well? Sorry, I can't see you from here.
Jordan Miller, Mayo Clinic.
I can't hear you.
Jordan Miller from Mayo Clinic. I don't know if this is working or not. Okay.
Name.
And so two questions. So first, with regards to aortic valve, have you gone back through your high throughput screening of aortic valve endothelium and tried to see if there's any evidence for epigenetic regulation of gene expression, specifically either repression of sirtuin enzymes or overexpression of DNA methyltransferases that might give you a clue that there's something similar, DNMT3A? Or?
No, in fact, that's my apology at the beginning. We're so hyper-concentrated on the arterial system right now, and just uncovering that's fresh for the arterial system, that we are looking exactly at what we're looking at SERTs, and we're looking at doing all the obvious things. But we have not, I've actually shrunk my lab a little bit recently. We haven't have a cohort of people working on the valve. What I think is, I think the future, and someone should do it in this room, should go and do some epigenetics on the valve. The exact point you're making. Because if those mechanisms also exist, and in fact KLF4 would be an excellent target in the valve as well, because it's so linked to eNOS and the possibility of nitric oxide regulation in the valve. So I think your question is exactly what needs to be done.
And I guess the second question being is integration of all these multiple levels of epigenetics, right? So you have, I think, famous last words, between the transcriptomics and methylation, that's a relatively easy one in comparison to the overarching layer of histone hyperacetylation and histone hypermethylation and whatnot.
Yeah,
so,
but you've got to keep one thing in mind. DNA methylation is at the forefront of all of the epigenetics, okay, because it regulates transcription. And a lot of the other regulatory pathways are secondary in a temporal sense to that. So I think that's where the emphasis, it's a decent place to put the emphasis at this point, to see how widespread this plasticity is, whether you're looking at arterial endothelium or looking at your favorite cell or looking at the valve.
Thank you.
Thank you, Dr. Jordan Miller. Any other questions? I'm sure there are a lot of burning questions with all this. Ismail? No?
You talked about mass transport.
Yeah.
Could you, rising interest in that area, could you enlarge?
Can you speak in the microphone?
So I can paraphrase, actually, if you like. So Colin is asking, what about mass transport? Because I did make a point of distinguishing it from biomechanics in the environment. It's there. You have to deal with it, but it's by definition is constantly changing, because as the cardiac cycle proceeds, the flow proceeds through different characteristics, and nothing is ever stagnant. And indeed, people who look at cells without flow and form conclusions and then give a flow to the cells are really comparing a no-flow situation, which for arterial cells doesn't exist except for a fraction of a second in the cardiac cycle. So all of our studies are compared with either in vivo or in vitro, are always with flow, whether it's disturbed or undisturbed, because that is reality. And I think we've been lucky in finding some very key molecules using a flawed approach, which is no flow versus flow. It's fine for looking at detailed cell biology of mechanotransduction, which I've certainly done many times over the years, because you're looking there for just a change, but it's not necessarily a good model for what's going on in vivo. Now, in terms of transport, again, that's the in vivo additive complexity, which we tend to ignore even in our in vitro studies, mainly because we don't have the tools to measure it as precisely as it's needed to be. Would you like to, you're the world's expert on mass transport, Colin.
Colin? Professor Caro?
I have to expostulate and say no. It's just that I think there is a fair amount of evidence around that, for example, oxygen transport is playing a role in atherosclerosis and in terms of intimal hyperplasia as well. And of course this links with the fluid mechanics, and it links with all your beautiful biology too.
Yeah. And the superoxide story we heard again this morning, and reactive oxygen species, all are important in transport issues as well as their immediate chemistry.
Any final question? Burning questions? I think you've convinced them.
Thank you.
David.